plant breeders take raw plant materials. They take cultivars that farmers grow or wild plants that have been collected, and they cross them together, and they look for progeny that they like, that improve the crop, that are better in color or better in shape or better in size or better in taste. They do a very, very basic activity of crossing A by B and selecting the best progeny. So my name is Erwin Goldman. I'm a professor in the Department of Horticulture, and I'm a plant breeder. I breed vegetable crops and try to improve them. I'm also a teacher. I teach several different courses in horticulture, evolutionary biology, plant breeding and plant genetics, and I also supervise students. If you wanted to start with a wild plant, and actually carrot is a good example because wild carrot would be Queen Anne's lace. And if you pulled up a Queen Anne's lace plant, you'd see that it had a kind of a white, fibrous, stringy root. You could eat it. I mean, it is edible, but you wouldn't really want to eat it. It's not very tasty. What we really want is this succulent orange or purple vegetable. And so that change from Queen Anne's lace to a succulent purple carrot or orange carrot represents thousands of years of breeding. The way we would do that would be to cyclically select in a population over time, every year choosing the best plants, recombining them, so intermating them, letting those plants interpollinate. In the case of Queen Anne's lace or carrot, we would put those plants together in a cage and put some insects in the cage, some bees or flies, let the bees or flies pollinate them, grow out the seed the next year, select the best plants, and do that year after year after year until we got the traits that we wanted to get. It helps for me to go back to the beginning of the crop. So this is a beet. This came from the Mediterranean. And if you go to the Mediterranean today, the coast, you'll find wild beets growing there, actually. So humans, I don't know, 6,000, 7,000 years ago, maybe the Romans, found that plant and started eating it. Actually, before they ate the roots, they ate the tops, ate the leaves. And over time, they selected something that had a slightly swollen root to it and began eating that thing as a beet. As they did that, they started to create different shapes and different colors, and it became a food product. And so in many ways, I think the tradition of domesticating the crop and making it into a food that people can reliably grow year after year is just something I'm continuing. This is a very interesting set of genetic material in our breeding program. We took a beet that's called Chioggia. A lot of people will know the Chioggia beet by it has red and white stripes. If you cut it open, it looks like a bullseye or a target. We crossed that red and white Chioggia parent with a yellow beet. And our goal was to try to make a beet that had yellow and red circles on the inside. And so that's what we're trying to do here. And we're not quite there, but we're getting closer. And so that's really those parents were crossed for that purpose. One of my favorite classical plant breeding stories is one that took place actually here in Wisconsin. It started here in Wisconsin with a farmer named Elmer Swenson. And Elmer Swenson was a farmer who was interested in growing grapes here, ultimately for winemaking. And he began to take the grapes that would have been typical of a European vineyard and select them over a very long period of time for winter hardiness. It's a very simple thing if you think about it. Putting those grapes out there, seeing which ones survive the winter, crossing the best, doing it year after year after year, and ultimately he developed winter hardy grapes for the upper Midwest. Those grapes went then to the University of Minnesota and to a couple of other breeding programs where they were bred into some pretty strong cultivars. Those cultivars are now the basis of the wine industry in the upper Midwest. We have over 100 wineries in the state of Wisconsin now. And it's largely based on grape varieties that were developed through classical plant breeding through a very simple technique of selecting in the environment in which you want to produce them. And I think it's a beautiful example of how plant breeding can not only modify these plant populations, but can also contribute to a local economy and to make, now I'm not going to argue that Wisconsin wines are yet competitive with other states, but the fact that we can do it here and that we even have wine tourism in the state and that we have ice wine, which we're quite good at making, is to me partly a plant breeding success story, and I think a really nice example of what plant breeding, classical plant breeding can do. Plant breeding and plant genetics are overlapping in many ways, but plant breeding is an activity that goes back probably 10 or 12,000 years as people began to domesticate crops, select the best things year after year after year. They were breeding it without really knowing anything about the genetics. And it wasn't until the work of Gregor Mendel in the 1860s and then the rediscovery of Mendel in 1900 that people began to understand that there was an underlying science really that underpinned the changes in those populations over time. So plant genetics would be the sort of the scientific underpinnings of what we're doing in selection by breeding. You know, in terms of the technologies associated with breeding, I mean, absolutely at the beginning it was very visual. We would choose the ones we liked, or maybe the ones that would taste good, and over time, especially as we learned genetics, we could then start to use tools, the tools of genetics or the tools of some of the more advanced sciences like biochemistry and statistics to make our selection much more efficient. So a way to describe that would be if I can select a plant at a very young stage, even when it's a seedling, if I can select based on its DNA, I might be able to know something about what kind of fruit that plant is going to make, but long before it's ever made a fruit. So that could make my breeding work much more efficient, and it could make my breeding work occur in a much smaller space and a much easier way than if I had to grow up every plant and look at it visually. So the new tools that plant breeders have are mostly tools that increase our efficiency and our ability to see into what we call the plant genome, the set of genes that make up that plant. It's sort of a window into that, making our selection more efficient. I might be able to do something in 3 years today that would have taken 25 years when I first started plant breeding 30 years ago. That's the difference that the new tools make. They make it more efficient. They make it much shorter, and in some cases actually they could decrease the costs associated with the breeding work, especially if you had to hire lots of people to do field work over 20 years compared to 1 or 2 people working in a lab in a very short period of time. So classical plant breeding is just based on crossing and selection. Genetically modifying plants, sometimes called transgenics or GMO type crops, really represent a very different approach to creating a cultivar in this one particular way. And that is that when I cross this carrot with this carrot, I am depending on the fact that those 2 carrots are closely related enough so that they will cross. But if I wanted to cross a potato with a carrot, no way I could make that cross. Nature doesn't allow that cross because those 2 plants are different enough that they will not intermate. Genetic modification is the ability to take a piece of DNA from anywhere in nature. I could take it from a bacterium, I could take it from a fungus, I could take it from a plant, I could take it from an animal, and introduce that into my carrot through a laboratory technique that would never happen in nature simply because that piece of DNA from a fungus would never interact with my carrot genes.
Genetic engineering opens up the possibility of more choices of genes to work with than traditional plant breeding, and that's why it's one of the reasons it's become an even enhanced tool for modifying crop plants, making crop plants better. So I'm Heidi Kepler. I'm an associate professor at the University of Wisconsin in the Department of Agronomy, and I work. My lab works on crop molecular genetics and breeding and genetic engineering.
So if you wanted to, let's say you wanted to introduce foreign DNA into a plant, you wanted to do transgenesis in the lab or create a genetically modified crop, you would need to isolate that gene from a bacterium or from some source, and you can do that through relatively simple laboratory techniques.
As far as identifying genes or candidate genes, often there's a trait, whether it's, let's say there's a trait in corn, if we're engineering corn, that we'd like to transfer, or if there's a trait in another organism that we would like to transfer to corn, you observe that trait, and then through different processes you isolate the protein that's been encoded to make that trait happen, and then by the sequence of the protein you can go backwards and isolate the sequence of that gene. And at that level, gene sequences that make genes are the same across organisms. So the way the base pairs work in fish is the exact same way it works in corn, is the exact same way it works in squirrels. They all use that same coding system when you get down to the gene level to make those proteins.
And then there are two main ways that you'd get the gene into your crop.
So the gene gun is one way to put genes, or what we call transgenes, into a crop plant or the cells of a crop plant. And it basically works in a sense like a shotgun, where you put your genes on the little particles. You can think of those like the BBs in a shotgun, and shoot those at high velocity toward the plant cells. And the DNA gets into those plant cells and gets into the nucleus and into the chromosomes of those cells and is integrated into the chromosomes.
A much more elegant way is the way that nature has been doing it for a very long period of time with bacteria. There's certain species of bacteria that infect the roots of plants.
Called agrobacterium, and this bacteria in nature, it exists, it's in the field, it's out there, and it naturally does genetic engineering. It's been doing it for eons, millennia, where it moves some of its genes into the plant cells and into the plant cells' genomes to get the plant cells to do the work for it, actually to make substrates that bacterium can eat and survive on. And so scientists over the years have been able to harness the power of that bacterium to selectively put the genes that we want moved into plant cells, to move those genes in for us in a more precise manner than the gene gun. Plant breeding is genetic modification.
There are many examples in nature of that phenomenon of transgenesis. So there are bacteria that insert their genes into plants. That's a kind of transgenesis. It's a kind of genetically modified organism. There are grafts that occur between roots of plants. That is another way of a plant inserting its genes into another plant. So it would be unfair to say that genetically modified organisms are something completely new that nature has never seen before. In fact, they do occur in nature, but they don't occur in a very sort of coordinated, directed framework where we're taking a specific gene from a bacterium and inserting it into a plant.
I think back to the base sequence and what makes a gene, and how this elegant system, because it works so well, has been utilized as the base of every living thing. And so this moving of genes and sequences from one species to another doesn't seem so foreign to me, because we've all been based on this type of sequence. Key benefits for genetic modification or genetic engineering is that we can, with this method, in a very defined way, add traits to crops or remove negative traits from crops in a way that's more precise than we've ever been able to before. And it's opened up the types of traits that we can add to crops beyond what we could do with traditional plant breeding. And so it's just opened up a whole realm of possibilities for improving crops. I just look at genetic engineering as another tool to approach the overall problem of how do we, in this day and age, what tools are available to produce the best crops with highest yields, least losses to pests and diseases, in a way that reduces the environmental footprint of that crop and makes it more sustainable, makes it a better practice on the environment than what we're doing now.
One of the greatest successes in the genetically modified crops world has been the transgenic papaya, which is a papaya that is resistant to the papaya ring spot virus, which is a devastating disease of papaya, completely destroys papaya orchards. Scientists were able to insert a viral coat protein gene that confers resistance to the papaya, and it has been an enormous success. Interestingly, activists have taken out entire orchards. People who are anti-GM have destroyed orchards of transgenic papaya, despite the fact that it has been, I think, largely viewed as one of the best successes of the technology in allowing crop production in an area where it should be produced, where if you had to rely on non-transgenic papaya, it wouldn't be successful. So I think that's an example of where this technology actually is playing a role in the world in a good way. I think there are three probably big areas why GM is of concern to people. One of them is that we're producing novel proteins in these plants that have never been seen before by nature or by people eating plants. And the possibility of producing a novel allergenic protein, or the possibility of producing something that could be harmful, is something that people are concerned about. It hasn't happened yet with our genetically modified crops, but I think that potential is something people are concerned about. A second issue is safety. In terms of environmental safety, you have genes that are now new genes that have been introduced into a natural landscape. Those genes can move from the crop into a weed. So using our example of carrots, if we had a genetically modified carrot, we don't, but if we did, that genetically modified carrot plant could introduce its genes to Queen Anne's lace, and that gene would then move out into the wild. So the safety of, and essentially the landscape and the escape of transgenes is something people worry about. And then a third thing would be the environmental concerns that people would have about some of the transgenes that are in use today. Allow for the use of an herbicide to control weeds. And while that's very effective for the farmer, it has meant a very large, wide-scale deployment of particular herbicides over millions of acres. And even though that is a part of our agriculture, I think there are questions about the wisdom of deploying herbicides like that over such a large area. In fact, we have dozens and dozens of herbicide-resistant weeds now that have been developing rather rapidly in response to these technologies.
And other people, it seems like they oppose not necessarily the science itself or the technique, but they don't like big companies. And because some of the first transgenic crops that were commercially available came from big companies, they've automatically jumped on that technique as being bad because they don't like big companies. And it's expensive. That's the other thing. It's very regulated. It's very expensive. And so it kind of locks out that technology for the smaller companies that want to try it and take advantage of it. Even here at the university, if we wanted to go and make a commercial transgenic crop, the cost of doing that is beyond our means, even at a big university like this.
There might be people who don't want the technology because they draw a line. I've had people say to me, I completely understand genetically modified technology. I think it's probably a safe technology, but I'm choosing not to use it, and I would like to choose not to use it, just like I'm going to make the decision to not use electricity, or I'm going to make the decision to take the Sabbath off, or I'm going to make the decision to draw a line of something that I want to do for my life. And so I found that response, and I think that's totally appropriate. A story that, and I think this is maybe more hubris than anything else, but I think it tells a lot about GM technology. Back in the early days of GM, a biotechnology company in California named Calgene had the idea to create a tomato that would be essentially able to be harvested ripe from California or Mexico, transported to the East Coast or to the Midwest and sold as a ripe tomato at any time of the year. It's a brilliant idea if you think about it, because the tomatoes that we get in the winter are often not so good. And the idea of having a ripe tomato would be very attractive if you were sitting in New York City in February. So to do that, they did a GM solution. They took the ripening gene of tomato out and they turned it around and reinserted it backwards. It's called antisense technology. And what it did was not shut down ripening, but it slowed it down. And it slowed it down in such a way that the tomato could be allowed to ripen fully on the vine. You could harvest the tomato, and then you could get it to market over 3 weeks, 4 weeks, and it would not rot. It's really a cool idea. And actually, there were many investors. Wall Street went crazy about this. People were like, this is going to be a huge thing. People invested lots of money in the company. It was one of the first times that plant breeding or seed companies had Wall Street visitors from Wall Street interested in investing, and it did what they said it did. It did allow for a harvest of a very ripe tomato. But Calgene was a biotechnology company, and they were a biotechnology company that had some brilliant people working for them, but they knew nothing about the produce business, and they knew nothing about plant breeding. So they took those tomatoes, they put them in trucks, they shipped them to Chicago where they had a warehouse, and they opened the doors of the trucks when they got to Chicago, and it was tomato juice in there. They had no idea how to ship the tomatoes, they had no idea how to handle the tomatoes, and they had only done the transgenic event in one variety of tomato. So anybody who knows anything about agriculture knows that farmers need 10 varieties. They grow different times of the year up and down the coast of California, they grow them in Mexico. So the plant breeders had not been consulted, and the people who do trucking and shipping had not been consulted. And I think it's a great example of the hubris associated with biotechnology. Brilliant people, fantastic ideas, even ones that work, but without the real full context of what it is we're doing. And I think in some ways I worry about biotechnology now, today, even in the same way that, yes, we can do these things, but what does it mean in the bigger context? Calgene, unfortunately, was not successful with their tomato, and it was partly because they failed to consider all of these bigger realities. But personally, I feel that the technology of GM so far has been safe for humans to consume. I do think the environmental concerns are real, but I think there's a place for that technology.
And people really trying to go in and find, bad things about transgenic crops are not finding it.
If you press me on scientific data to say that it's bad and completely should be banned, I could never do it because the data don't exist. I think wholesale just exclusion of the technology as though there's something really horribly wrong with it is a bit misguided.
So why aren't people fully supportive of GM or GE crops? Sometimes I wonder, when I've talked to people, sometimes it's just that they're totally misinformed about what the process is, what it does, the accuracy of the genetic engineering process, and so they're told in an inaccurate way about this technology, and they become scared. But they're told a lot of scary, bad, totally untrue stories.
So far, we have not seen a genetically modified crop that's been released to the market that has clear, safe health risks. However, we do have to be diligent in looking for the possibility that any novel allergenic protein could be there. Secondly, with respect to the escape of transgenes into the wild, I think that has been demonstrated. In fact, there are some studies that were done in the West Coast that showed pollen from transgenic grasses could move, I think they were moving 20 kilometers on the wind. So clearly we have to expect that whatever transgenes we put out there will likely move into the environment. And third, the environmental concerns that people have raised that relate to herbicide-resistant weeds, I think we're seeing that already, and resistant insects. So these are all things that would occur under regular typical agriculture anyway, but they're occurring under a much more rapid rate with transgenics.
And so I'm frustrated that there are, in each area, there are some really polarized thoughts and groups that I think there's good ideas kind of in all of those camps, whether it's the conventional sustainable, organics, molecular genetics, and that we could all come to the table with kind of putting together the best ideas. What if we're going to, build the ideal crop or cropping systems? What are the tools and what are the ideas that we can pull together and kind of leave the politics out of it and go with that approach? And sometimes it's really hard to get people in different camps to come to the table. They tend to close their ears and eyes and not want to be open to at least discussing possibilities.
I actually think that there will ultimately be a subset of, and it may not be officially organic because organic may never allow it, but I think there will ultimately become not bifurcated but just stratified system where there's more sustainable methods that do include those technologies, and then there will be sustainable methods that don't. And I think it will change over time, simply because I believe there probably will be transgenics that will have a really strong positive environmental benefit, or will have a really strong nutritional benefit that will make sense for people.
So as a scientist that's working in research of transgenic crops, I would say to the general public, don't be afraid of the technology. To learn more about the process and to understand it's not a fearful new technology. I think new technologies in general, people just, it's change, and it's sometimes hard to understand. But to go to literature, and not the biased literature, or go to universities, have talks, online, try to find unbiased sources, but the information is out there showing the proven safety of transgenic crops. I hope, and it seems to potentially be going this way in the future, that some of the polarized camps are going to come together and realize that this technology, along with other technologies and information that we've developed, can be utilized together to make safe, productive, environmentally friendly crops of the future.